Benedict's test is a chemical test that can be used to check for the presence of reducing sugars. Reducing sugars are aldehyde sugars or aldoses. Monosaccharides that have aldehyde functional groups are reducing. Disaccharides may be reducing if they have a free aldehyde group in the form of hemiacetal, or non-reducing if they do not have a free aldehyde group. They have acetals carbons. In this test, Dr. Emilia will use the experiment described in the manual to show you the results of Benedict's test. After that, she will use hydrochloric acid to hydrolyze sucrose into its constituent monosaccharides, glucose and fructose. This will be detected using Benedict's test because sucrose as a disaccharide is non-reducing, but one of its monomers, glucose, is reducing.
Here we have the results for the Benedict test. We are seeing positive result if there is a reducing sugar and negative result if the sugar is not reducing. So it doesn't have anything to do with mono or disaccharide, just with the reducing or non-reducing. Here we have 2% glucose. You see clearly the formation of a brownish red precipitate, which indicates positive result. The same with lactose, maltose. Fructose is the prettiest, very red, and so these four are positive. Sucrose is negative, starch is negative, and our negative control, water, gives the same color, as you can see, as the starch and the sucrose, yeah? While you can see very clearly the difference between water and fructose, water and glucose, yeah? So that's our result for the Benedict's test.
In the hydrolysis test, similar to the way you make sugar syrup, cutter, sucrose is added to a test tube with HCl and boiled for a few minutes. The acid will hydrolyze the glycosidic bond between glucose and fructose.
Okay, the 2 minutes are finished. Now I take them out. Hopefully the HCl has broken the glycosidic link between the disaccharide. I'm going to add the reagent that is going to show me whether I'm dealing with the disaccharide or not, or with the reducing sugar or non-reducing sugar, which requires, as you remember, putting it in the water for 2 to 3 minutes. Okay, this is the result after adding Benedict's test. Now in your manual it says Barford's test, but we did it with Benedict. You seen before Benedict had a negative result when we were dealing with the disaccharide sucrose, but positive result with the monosaccharide. And here is the sucrose with the HCl is positive, while the sucrose to which we just added water, and so we didn't do hydrolysis, stayed as a non-reducing disaccharide. So here is your positive result. Sucrose here is no longer sucrose. It has been hydrolyzed to 2 monosaccharides, glucose and fructose. Okay.